Good afternoon, dear judges and the participants of Concours. I'm Beridze Raimond Anzorovych, the pupil of the Mark Young Scientist Ecological Research of the KPNZ Odessa Ecological and Naturalistic Center Afalina. I want to present to you my scientific research, study of the influence of coniferous plant extracts on the growth and development of wet and reddish seeds in vitro and in vivo, and the development of physiologically active drugs. Physiologically active substance of coniferous plants that grow on the territory of Ukraine in parks, forest stripes, and forest massifs can be used to create stimulant and herbicidal drugs that would be safe for humans and the environment. Also, herbicide preparations from conifers can replace traditional herbicide preparations in the long term, as they do not cause adaptation in weeds due to long-term use because they contain a wide range of chemical compounds. Traditional herbicides can also cause various diseases, including cancer. Therefore, the aim of our work was to study the effects of physiologically active substance or of extracts of yewberry, virginian, juniper, Himalayan pine, black pine, cedar of Lebanon, and cedar of Atlas in vitro and in vivo to create physiologically active drugs. The objective of the study were to conduct a series of spring, summer, autumn, and winter experiments to analyze the effects of coniferous plant extracts on the growth and development of roots of reddish variants, national and sexa, and wet variants, Arnautka and Luzanivska in 2023 and 2024. Comparison of research in 2024 in Odessa and in Nizza. Study of the effects of coniferous plant extracts in vivo, analysis of the data obtained and their application for the production of herbicidal and stimulating drugs. The work was carried out in the Yunatsky Park Monument of Landscape Art of the Regional Humanitarian Center and the Afalina Ecological and Naturalistic Center in Odessa in 2022-2024. In our work, we used the basic methods of extraction of coniferous tree on the water base and also the standard biotesting methods. Also, to analyze the results of the study, we used the standard mathematical methods of analysis. We conducted more than 500 experiments in 2024 to study the effect of extracts of yew, Himalayan pine, black pine, Lebanese cedar, Atlas cedar, and a mixture of Atlas cedar and Himalayan pine in different concentrations. In the spring experiments in Nice, the root length of wet Luzanivska germinated on coniferous plant extracts in March and April was shorter than the control in all experiment variants. The effects of coniferous plant extracts from Odessa different from the effects of extracts from Nice. The data from Odessa and Nice were analyzed using t-test. Unlike the extracts from Nice, the extracts from Odessa in March had a stimulating effect in 6 out of 10 variations. We can observe it on the graphic on the figure 2. In April, the extracts of conifers had a stimulating effect in 2 out of 10 variations. We also can observe it on the figure 2. The strongest stimulation effect in March was shown by the extracts of the mixture of Atlas cedar, Lebanese cedar, and Himalayan pine at a concentration of 1 to 7. Root stimulation in comparison with the control was by 32%. In April, the strongest stimulating effect was also shown by the extract of the mixture at a concentration of 1 to 7. Root stimulation was by 14%. In the summer experiments in Nice, the length of the roots of wet germinated on the extracts in June, July, and August was less than the control in all experiment variants. We can observe it on the figure 3. The extracts of conifers from Nice in summer had a stronger inhibition than the extracts from spring. The highest inhibition was in the extract of yew berry at a concentration of 6 grams per 100 milliliter. In 2024, we conducted a number of experiments to identify stimulant, adaptogenic, herbicidal, and retardant effects in vivo. We conducted experiments to test extracts of a mixture of Atlas cedar and Himalayan pine at a concentration of 1 to 10 on the growth and adaptation of Aleppo pine seedlings. We also conducted experiments on growing yew seedling using an extract of a mixture of Himalayan pine and Atlas cedar at concentration of 1 to 10. Also, we used a mixture of Himalayan pine, Atlas cedar, and Agro mineral fertilizer, 50% of the mixture and 50% of the fertilizer. Our experiments showed that the extract of the mixture and the extract of the mixture with mineral fertilizer stimulated shoot growth by 30% compared to the control where shoot growth was 10%. Referring to our in vitro and in vivo studies, as well as our microscopic studies, we proved that yew-berry extract inhibited the stretching zone of wet root cells. We can state that yew extract can be used to create a herbicidal or retardant preparation. In experiments comparing yew-berry extract and the herbicide Roundup, yew-berry showed the same effectiveness as the herbicide. Our in vitro and in vivo studies have shown that the extract of mixture of Atlas cedar and Himalayan pine, as well as a mixture of Atlas cedar, Lebanese cedar, and Himalayan pine, can be used as a stimulant or adaptogenic preparation. The extract of a mixture of Atlas cedar, Lebanese cedar, and Himalayan pine at a concentration of 1 to 7 in March stimulated the growth of seed roots of wet variety Luzanivska. Our long-term in vitro and in vivo studies to identify the physiological effects of coniferous plant extract allowed us to develop our own permanent herbicidal and stimulating drugs from yew berry and a mixture of Atlas cedar and Himalayan pine. This preparation has an average density level and can be stored for 3 to 6 months at a temperature of 4 to 8 degrees Celsius. Thank you for your attention.